Hello, this is Miss Dagenford, and I need to redo these videos. The sound was quite bad on the other ones. So here we are with the intro to photosynthesis. So here's the overview and the basics, and you can see we start with the leaves. We already did the cross section of the leaves, but just to refresh your memory, here's the top layer, the palisade. The palisade level, that's where a lot of um, the the main portion of photosynthesis occurs as the light comes streaming down on top. Not always, depends on the plant. Below that, we have the spongy layer, so called because of the looser arrangement of the cells. There's more air spaces and allows for the exchange of the gases. If we pick one of these if we pick one of these cells and enlarge it, we can see the chloroplasts inside of them, the little green organelles. And if we magnify those and kind of use an artistic version of it, we can see that the chloroplasts have two main portions. We have these green stacks of membranes called thylakoids. Strange name. These thylakoids are where the chlorophyll is located, and we're going to see that's where we have the light dependent reaction. Anything that surrounds the thylakoid, all of the surrounding area, that's the stroma, and that kind of bathes or surrounds those stacks. Um, and then you have the inner membrane and outer membrane. If you recall, this is an organelle that is analogous to mitochondria. So we're going to see the double membrane. We talked earlier about the endosymbiotic theory, where these organelles are theorized to have come from prokaryotic or cells that were consumed by other cells, and then they stuck around. This was the equation we talked about in class. We just hinted at it. You should remember that in photosynthesis, carbon dioxide is consumed, glucose is produced, as well as oxygen. That's what is released. You require water as well. Now, in the history of the discovery of photosynthesis, one of the things that they wanted to figure out is, well, what reactant is being used to produce each product? And so they used isotopes that were traceable. And this is what they found, that the carbon dioxide, which this one made sense. A lot of, I don't think this came as a surprise to anybody. The carbon dioxide was being used to produce the glucose. Um, Now, the big question was what's being used to make the oxygen? And when they labeled the oxygen, when they used an isotope, they were able to determine that the oxygen gas came from the water that was being used, not the carbon dioxide. And then they found that the hydrogens were being rearranged between the glucose and water. And then the oxygen in carbon dioxide is also used in the glucose. So nice little reminder that if we want to determine something, we can find something that allows us to trace it. We've talked about those experiments before in class. So this is a very nice schematic. You're going to notice the two main reactions. Now this is a schematic, so this is just a nice symbol. Um, Obviously, the light reaction is not just over on the left, and the light um, independent is not just on the right, but it's a nice way to organize it. So over here, we do have the light dependent reaction, and then over on the right side, we have the light independent reaction. And here we have our thylakoids, and we have light shining down. Let me get a nicer pen here. There we go. And that's why it's called the light dependent reaction. And what you'll find is we have water coming in, and in the process of the light dependent, we're going to produce our oxygen. This is going to be our wasteful gas. And we're going to produce two energy products. Very important here. We're going to have ATP and then a molecule called NADPH. That should sound somewhat familiar to you. When we did cell respiration, we saw NADH. They're going to be used in the Calvin cycle also called the light independent reaction. And in the light independent reaction, we're going to take carbon dioxide, and through the reactions in the Calvin cycle, we're going to generate our glucose. Or a little bit more, oops, that's O6, sorry about that. A little bit more accurately, we're going to be first producing a 3-carbon sugar, but that can then be used to make glucose, which can then be used to produce starch, or fructose for fruit, or sucrose for transporting around the cell. So the plants can do anything at once at this point with this glucose. Nice little review point: when you combine these two molecules together, we're going to condense or pull water out in that dehydration synthesis to create our disaccharide. And just a nice refresher for them. So and in the process of the Calvin cycle, we're going to regenerate NADP with a positive charge, and then ADP because we've used up the energy, and then our phosphate. So this is a really nice energy transaction. The plants can just happily stand underneath that sunshine and photosynthesize all day long. So there's our diagram in a nutshell. Before we get to the details, though, we need to go over pigments and light energy. And so we need to refresh our minds with the visible light spectrum. Over in the purples and blues, we have shorter wavelengths and higher energy. And then over on the reds, we have longer wavelengths and lower energy. And then green and yellow and orange, they're right in between. What we find out if we use the spectrophotometer, now spectrophotometer is a machine that allows us to measure how much light wavelengths are either transmitted or absorbed. And you could also use a colorimeter for this. And what we notice with green, so notice here we're using a prism and only using green wavelengths. Well, what this is indicating is that it's being transmitted. In other words, the green wavelengths are going right through the pigments. Now, the take-home message of that is plants don't absorb. That's a do not. There we go. Plants do not absorb green wavelengths. Um, Now, if we use something like blue wavelengths, pardon me, if we use our blue wavelengths, there we go, then we find that the plants absorb it. Now, so notice the needle is down at 0. The plants like blue wavelengths. So in other words, we see that plants being green, that means that the plant pigments are reflecting and transmitting the green wavelengths, and they're absorbing primarily the reds and blues. There are some other pigments that can absorb some other wavelengths, but predominantly it's red and blue. Those are the chlorophyll molecules. So if we do absorption spectrum, where we kind of look at what pigments like what wavelengths, we have chlorophyll A and chlorophyll B. They're both green. They're slightly different shades of green. One's a bright green, one's a bluish green. And then we have carotenoids, which can absorb a little bit. You'll notice they kind of get into the blues and the greens, but not much. Um, you'll notice again that plants by far, they're not absorbing the greens and yellows, and they like the reds and blues. Now, Engelmann, a good century ago, Engelmann was a scientist who did a very clever experiment. He took a little filament of algae. And algae, being photosynthetic, algae are producing oxygen. Well, no problem there, no biggie, as it's producing oxygen. But what he did is he used a spectrum, not a spectrum, I keep saying that, a prism, sorry, a prism to separate the light wavelengths. And he also then used aerobic bacteria. Now, aerobic bacteria would require oxygen. And as it requires oxygen, it's going to then grow very well where there is oxygen. So he, even before all these fancy machines were invented, he had a very clever experiment that kind of showed that the bacteria are growing where there's the most oxygen. And there's the most oxygen because there's the most photosynthesis occurring in the reds and the blues, and approach a little bit of purples, but that's where we're seeing a lot of the bacteria growing. Very clever, very intuitive little experiment. And then when we had the technology, we were able to kind of look at that and say, oh, now we see why we're getting that curve. The chlorophyll A, chlorophyll B, they're absorbing these wavelengths that mimic, that allow for that photosynthesis to occur. Now, one last thing for this video is simply the main um, pigments involved are going to be chlorophyll. So this is a structure of chlorophyll, and just as a nice reminder for chemistry, notice here we have our hydrocarbon chain, our tail, that's going to be hydrophobic. So nice application here. This is what's going to be going into the membrane. So here we have our little lollipop structure. There's the tail of the chlorophyll, and then here's um, the little ring that has the magnesium at the center. I had asked a question in an earlier video, which one of these pictures best represents where chlorophyll would be placed in a membrane. And it can't be this one because you only have one hydrophobic portion. And here this would be hydrophilic, this would be hydrophilic. It's not going to be this one because this is hydrophilic, it's polar, it's not going to be in with the nonpolar tails. And then we have our uh, letter B here. That would be the best model that shows where it would go in the membrane. So the idea of a pigment, pigments can absorb wavelengths. And in the case of chlorophyll, we have a molecule that absorbs those reds and blues. And what you're going to find in the membrane is that we have these little reaction centers that allow for the collection and harvesting of light energy. So here's our thylakoid membrane, and our last little bit for this video will be just to show you that we have these proteins. The purple is the protein, and then we have these little chlorophyll, all of these chlorophyll molecules that are absorbing light wavelengths. And what they can do is they can harvest, they can absorb the energy, then transfer it. And then they're going to transfer it to a special pair of chlorophyll molecules that can actually then give up that energy within that electron. An electron gets so energized that the electron pops out and can be used, that energy can be used. Well, hope that helps, and hope this is a better sound quality than the previous one. Thank you.